everyone, I am Dr. Sonal Jain from Department of Anthropology, University of Delhi. In this series of forensic science lecture, we are going to discuss about various immunochemical techniques. There are certain tests in immunochemical techniques which we can perform to identify various forensic biological samples which are found at the crime scene. These techniques are most useful in forensic serological cases where we have to identify species identification, blood identification, semen identification and other biological substances which consist of antibodies and antigens. There are various techniques which can be used for analysis for such precipitation reactions, agglutination reactions, complement fixation, ELISA, radioimmunoassays, etc. These techniques can be qualitative in nature as well as quantitative in nature. Mostly whenever we are going for immunochemical techniques, we are talking about antigen and antibodies. Whenever any homologous antigen and antibodies combine together, they form a precipitate and we call it as a precipitation reaction. These precipitation reactions are useful for identifications of various antigens as well as various antibodies. And we perform various tests for identification of antigens and antibodies. These techniques can be qualitative in nature as well as quantitative in nature. So, we have learnt about the ring test in our previous lectures. We are going to start with the slide test. As the name suggests, the slide test can be performed on a slide. A drop of antigen and antisera is placed on a slide and we mix them by shaking the slide. If the presence of follicles is there on the slide, then it confirms that the antigen and antisera or the antibodies are of the same species. For example, we can confirm about the VDRL test, RPR test for syphilis using that. The most common test which we can perform in the forensic cases on a slide test is the blood grouping test. For example, any blood is found at the crime scene and the first step is where we have to identify that this blood belongs to which blood group. For example, we have A positive, B positive, AB positive, O positive, O negative, A negative, B negative, AB negative, different blood groups we have. For blood grouping, we go for the slide test. Whatever blood we have found at the crime scene, we will put that blood on the slide and then we have different antiseras. Whenever the particular blood coagulates or produce floccules with the particular antisera, then we can confirm the blood group of a person. The AB blood group person will give positive reactions with both A antisera as well as B antisera. And for the positive factor, Rh positive factor, we have to put another sera to know what is the blood group of the person. So, slide test is very important for the blood grouping, for the RPR test for syphilis. Tube test or the ring test is employed for the standardization of toxins and toxoids. Serial dilutions of toxins are added to the tubes containing a fixed quantity of antitoxin. The amount of toxin that flocculates optimally with 1 unit of antitoxin is the LF dose, that is the Khan test for syphilis. Basically, here we are talking about the quantitative reaction. In the tube test, we can take multiple tubes in a rack and then we apply in the put the serial dilutions of a particular toxin, and depending upon after how much addition of toxin the flocculation is happening, we can know what is the 1 unit of the antitoxin after which the reaction will occur. So, ring test is there, slide test, tube test. Now immunodiffusion is another type of immunochemical technique. In immunodiffusions, the reactions is visible as a distinct band of precipitation. Immunodiffusion reactions are very stable and they can be stained for preservation. They indicate identity, cross reactions, and also the non-identity between different antigens. If there will be no reaction occurring, then there will be no non-identity between different antigens. Whenever we are talking about the immunodiffusion technique, the immunodiffusion technique can be divided as single diffusion in one dimension, single diffusion in two dimensions, double diffusion in one dimension and double diffusion in two dimension, and also immunoelectrophoresis, where we have to apply an extra electricity for the reaction to happen. In single diffusion in one dimension, this is also known as the Audin procedure. Antibody is incorporated in agar gel in a test tube and antigen solution is layered over it. As we can see in the image below, that the agar is put which has antiserum that reacts with two antigens, and on the top of that we have put antigen with very high concentration. In single diffusion in one dimension, as the antigen is in high concentration, then it will move in one side only towards the antibody, and the reaction will be formed in the pre-zone. As we have discussed in the previous lectures about the pre-zone, pro-zone and the zone of equivalence, whenever the antigen is put with a high concentration, the reaction will go in single dimension downwards towards the antibody, and in this case we have put two antigen solutions. So we can see two antigens or two bands with two different molecular weights. That means reaction has happened with two different antigens in two different molecular weights. This is known as single diffusion in one dimension, also known as Audin procedure. Going towards the next procedure, we have double diffusion in one dimension. It is also known as Oakley-Fulthorpe procedure. Antibody is incorporated in agar gel. On the top of that we place a column of plain agar and the antigen is layered over it. Now the antigen and antibody move towards each other through the intervening column of plain agar and then they form the precipitate. In the double diffusion reaction, the reaction will be happening from the two sides. That's why it is known as the double diffusion reaction. So we have antibodies in the lower part of the tube, we have visible precipitates in between, and we have antigens in the upper part of the tube. So the antigen are moving towards the antibodies are also moving towards the antigens, and then the reaction is happening in the between, which is known as the zone of equivalence. As the amount of antigen and antibodies both are similar, the precipitation will be formed in the between. The visible precipitates will be seen at the center of the tube, but the reactions are happening from the both sides. Antibodies are going towards up, antigens are coming towards down. So we call it as double diffusion reaction, because the precipitate is formed in between agar gel layer where nothing is present. Antibody has moved towards up, antigen has come down, and the precipitation is formed in the zone of equivalence. The same thing is happening in the case of ring test, which is a double diffusion in one dimension, where a single precipitate is forming in between. Now the third part is the single diffusion in 2 dimensions, also known as radial immunodiffusion. In this technique, the antibody is incorporated in a gel and it is poured on a flat surface. Wells are cut on the surface to which antigen is added and it diffuses radially from the well and they forms ring shaped bands of precipitation concentrically around the well. So, what we have done in this technique? We have taken gel, agar agar, and we have directly put antisera in that gel. Now, we have made a gel with antisera in it. After that, in a open tube we can take, in an open dish we can take. After that, with a hole puncher, we will cut the wells in that gel and in that holes we will put the antisera. Now, the whole gel is with the antibodies and in the wells the antigen is there. The antigen will diffuse towards each side because it is surrounded by the antibodies. As it will diffuse towards all directions, that's why we call this technique as radial immunodiffusion, because radially it is spreading all over. We can see in the image that how in the open dish with the gel, the antisera is being put, wells are cut in that, and as you can see the white reactions in the lower image and in the upper image, as you can see the red lines, which is confirming that antisera and antibody reactions, flocculation is happening or agglutination is happening depending upon what kind of reaction is that. One more thing to consider, if you look at the upper image, that the amount of precipitation or the reaction in the first ring is very much compared to the last ring. That means the amount of antigen put in the first was, the concentration was very high, then it was lower, then it was lower, and again then it was a lower. So, what do we call such reaction? As radioimmunodiffusion or single diffusion in 2 dimensions. Now, the next is we have double diffusion in 2 dimensions. Double diffusion in 2 dimensions is also known as Auchterlonie procedures. It helps to compare different antisera and antigens directly. Agar gel is poured on a slide and wells are cut. Antisera is just put in the central well and different antigens in the surrounding wells. So, what we have done in single diffusion in 2 dimensions, we have mixed the antisera in the gel and in the wells we have put the antigens. But here the gel is pure, we have mixed nothing in it, but what we have done, we have made a central gel well and put antisera in this, and in the surrounding wells of that we have put different antigens. Now, how the reaction will happen? So, in the central well where the antisera is put and in the surrounding well where the antigen is put, we can see the lines. If the lines are very thick, then we can say that it is an identical reaction, we can identify the substance. If the reaction is not there, then we can say lack of relatedness, and in the third we can say it is a partial identity. So, a V-shaped mark is formed in such reactions. In such kind where LX gel preparation, it's a similar kind where we can put the antisera in between and the antibodies in the outside wells, and then we can see the precipitation lines. Now, after talking about the single diffusion in 1 dimension, double diffusion in 1 dimension, single diffusion in 2 dimensions, double diffusion in 2 dimensions, we have come for the immunoelectrophoresis techniques. As the name suggests, this technique is a combination of immunochemical techniques as well as electrophoresis technique. In electrophoresis, we use an apparatus where we have buffers, a agar gel and a current to support our reaction. So, this is a combination of immunochemical technique plus electrophoresis technique where we will apply the outside current for the reaction to happen. Graber and Williams were the scientists who have developed the technique of immunoelectrophoresis. As the name is suggesting, it involves the electrophoretic separation of composite antigen into its constituent proteins, followed by immunodiffusion against its antisera, which will show separate precipitin lines. Now the question arises, whenever the reaction of an antigen and antibody is a reaction which is self happening, why do we need to put the electrophoretic apparatus to that? So, in certain cases, in the cases of antigens, it is combined of various proteins and we have to identify different proteins or the different components or the different constituents of the antigen. So, the electrophoretic apparatus will help us or help the antigen to identify or to separate into its different constituents, and then the immunodiffusion reaction will happen with the antisera of these antigen reactions, which will show different separate precipitin lines. This immunoelectrophoresis, it is performed on an agarose gel with an antigen well and antibody trough cut on it. The test serum is placed in the antigen well and electrophoresed for about 1 hour. An antibody against human serum is placed in the trough and diffusion is allowed for around 18 to 24 hours. So, when are we putting the antigen into the well and we are electrophoresing it for 1 hour? So, it will separate into its constituents, and near to the constituent we will put different wells antisera trough, and we will give around 18 to 24 hours for the immunodiffusion reaction to happen. So, in such cases the electrophoresis is happening before and the immunodiffusion reactions are happening later. So, the antigen is dividing itself into various constituents, and these constituents, when they are separating, there in the trough we have put the antisera, and as you can see the lines which have developed as a result of the reaction between the antigen and the antisera. The last image is showing the curved lines, the half semicircle lines, which is the reaction between the constituents of the antigens and the antisera which is placed in the trough. So, the reaction line is shown by the precipitation line between the circle antigen and in between the trough which is present where the antisera is being put. So, positive reaction is determined by these lines. That means the antisera is showing a homologous reaction towards these antigens. This is also immunoelectrophoresis image where these lines are showing the reaction. Also, the intensity of the precipitation reaction shows the affinity. That means they are more homologous to each other. So, that is why the precipitin which is being formed, the agglutinin which is being formed is very strong compared where there is lightly formed. So, electroimmunodiffusion, it is the development of precipitin lines which can be speeded up by the electrically driving the antigen and antibodies. We have two types of electroimmunodiffusion: counter immunoelectrophoresis, one dimensional double electroimmunodiffusion, and rocket electrophoresis, which is one dimensional single electroimmunodiffusion. Whenever we have to speed up the process of immunodiffusion, we can speed up the process by applying the electrical devices. So, there are two types of those electrophoresis. One is counter immunoelectrophoresis and the other one is rocket electrophoresis. In the counter immunoelectrophoresis, also known as CIE, it involves simultaneous electrophoresis of antigen and antibody in gel in opposite directions, resulting in precipitation at a point between them. So, what we do in a counter immunoelectrophoresis, we have made a gel, and on the opposite ends we have put antigens and antisera. On the one end of the gel we have put antigen, and on the other end of the gel we have put antisera or antibodies, and we put the gel in a buffer, and then we have put one electric device. With the help of the electricity, the reaction will be speed up. Otherwise, the antigen and antibody in gel will travel with a slow speed, but whenever there is a buffer present which will provide the electricity, will provide the current to the buffer, and it will help the antigens and antibodies to move towards each other, so that the reaction will be speed up. If the electricity is not provided, the reaction might take 18 to 24 to 48 hours, but with this counter immunoelectrophoresis procedure, the precipitation lines can be visible even within 30 minutes. It is used for detecting antigens like alpha fetoprotein in serum, antigens of cryptococcus and meningococcus in the cerebrospinal fluid. So, a precipitin band is formed between the antigen and antibody reactions with the help of the buffer and the electricity. The antigen and antibody, with the help of electricity, they move with a speed up in the tray, with the speed up reaction and with the high speed, so that the band is formed within a less time. The second part of electroimmunochemical technique is the rocket electrophoresis. It is used for the quantitative estimation of antigens. The antisera to the antigen, which is to be quantified, is incorporated in agarose gel on a slide. Antigen in increasing concentrations is placed in wells punched in the said gel, and the antigen is electrophoresed into the antibody containing agarose. The pattern of immunoprecipitation resembles a rocket, that is why we call it as rocket electrophoresis. So, what we do in this rocket electrophoresis, this is basically a technique for quantitative examination. The antiserum or the antibodies which are to be quantified is incorporated in the gel only, like we have done in the previous cases where radioimmunodiffusion. Similarly, whenever we are making an agarose gel, we are putting the antisera in the agarose gel, and in those gel we will make particular holes. Supposedly, we consider a rectangular gel we have make, and we have mixed the antisera in that gel while preparing the gel. After the gel is set, we have punched 5 holes on the top, and in each hole we are increasing the amount of antigen. In the first hole we are putting 1 ml, just for the case we are putting 1 microliter, then 2 microliter, 3 microliter, 4 microliter. We are increasing the quantity of the antigen in the each well. Now we say in the whatever the in which reaction the quantity of antigen is very high, the reaction will be a highest peak, and where the quantity of antigen is low, the reaction will be a lowest peak. Looking at the image, we can clearly see that in the sixth well, the peak is very high. So the amount of antigen in that reaction is the highest. So supposedly, if we are identifying any toxin in a sample which is being collected from the crime scene or which is being provided, and we have to know the amount of toxin present in that particular sample, we can make the dilutions of the toxins in the different wells, and then we will be able to know that which well contain the highest amount of antigen in that. And in this case, the antisera is already mixed with the gel. Now, for the variant of rocket electrophoresis is the Laurel's two-dimensional electrophoresis. The antigen mixture is electrophoretically separated in a direction perpendicular to that of the final rocket stage, and it is used to quantitate each of the several antigens in a mixture. So, if we have to do single, we can go for the rocket electrophoresis, and we have to do double, then we go for the Laurel's two-dimensional electrophoresis, where the antigen mixture is electrophoretically separated in a direction perpendicular to that of the final rocket stage. So, by using the techniques which we have discussed, depending upon what kind of variation we want in our immunochemical reactions, whether the reactions can be conducted in a test tube or in a slide, or they require an electric force from the outside, we decide which technique we have to use for that particular sample to be analyzed. So, in conclusion, we can say that the technique depends upon truly the reaction which we are looking for, the sample which is present, and the result which we are looking forward to. And these techniques can be used in some cases as a presumptive test and in some cases as a confirmatory test, whose report is directly submitted to the court of law and can be applied in the court of law. Thank you.